Hello, Dr. Bohinski. My name is Ariel Hill, and I will be presenting Progress and Prospects, Zinc Finger Nucleases as Gene Therapy Agents, a review of ZFN technology and therapeutic gene modification. Gene therapy aims to treat or prevent disease by correcting or modifying faulty genes within the patient's cells. Precise gene editing is essential for treating genetic disorders caused by specific mutations, ensuring only the target gene is affected. In gene therapy, traditional methods face challenges like random DNA insertion and low correction efficiency, highlighting the need for tools like ZFNs for accurate targeted gene repair. ZFNs are engineered proteins that combine DNA-binding zinc finger domains with the foci nucleus. Each zinc finger recognizes a specific DNA triplet, allowing precise target site selection. ZFNs then create double-strand breaks, known as DSBs, enabling targeted genome editing via DNA repair mechanisms. ZFNs induce DSBs at specific genomic locations. These breaks activate two main repair pathways known as non-homologous end-joining and homologous recombination. Non-homologous end-joining is error-prone and can disrupt gene function, where homologous recombination enables precise gene correction using a donor template. This mechanism allows for both gene disruption and correction. The advantages of ZFNs for gene therapy are that they enable precise targeting of specific genes, they are more efficient than traditional homologous recombination alone, and they allow correction of disease-causing mutations or selective gene inactivation. ZFNs were tested in human cell lines and animal models. They were used to correct or disrupt genes such as CCR5, which is linked to HIV resistance, and they were measured by their cleavage efficiency, gene modification rates, and off-target effects. ZFNs and HIV gene therapy, real-world impact. Now that we've looked at preclinical applications, let's move into a real-world example where ZFNs have shown therapeutic potential, HIV treatment through CCR5 gene knockout. The CCR5 receptor is a key entry point for HIV into CD4 plus T cells. By using ZFNs to disrupt the CCR5 gene in the patient's own T cells, we can make those cells resistant to infection. In a phase 1 trial by Tabas and colleagues in 2014, participants saw up to a 40% reduction in viral load, and importantly, the modified cells persisted for over a year. One participant was even able to maintain stable CD4 counts and lower viral load without continuous antiretroviral therapy. Compared to newer tools, ZFNs have an established safety record. The CRISPR-Cas9 is easier to design but can carry higher off-target risk, and TALENs, while flexible, haven't been used as extensively in HIV trials. This example illustrates how ZFNs remain clinically relevant in today's rapidly evolving gene editing field. The results concluded that ZFNs increased gene editing frequency significantly over baseline. There were successful correction of genes in both dividing and non-dividing cells, and some off-target effects were observed, but generally manageable with careful design. Challenges and limitations. While the results are promising, there are still significant barriers to widespread clinical use. Off-target cleavage can cause unwanted mutations, and designing highly specific zinc finger domains remains complex and time-consuming. Delivering ZFNs efficiently into target cells, whether through viral vectors or electroporation, can also be challenging, and each method has its own risk, such as immune responses or genomic instability if edits are repeated. On top of that, these therapies are expensive, which limits access in low resource settings. Understanding these limitations is essential before moving ZFNs into broader clinical practice. Ethical considerations in ZFN-based gene editing. In addition to technical challenges, there are important ethical questions. First, the distinction between somatic and germline editing is crucial. Somatic edits affect only the treated patient, while germline edits could be passed down to future generations, raising long-term societal implications. We also have to address equity of access, as the high cost of gene therapies could worsen global health disparities. Finally, informed consent is an ongoing challenge because these technologies are complex and patients must truly understand the risks and benefits. Balancing innovation with public trust will be critical as we decide how and in whom these powerful tools should be used. Some current improvements and engineering efforts are that the modular and selection-based methods improve zinc finger specificity. The use of paired ZFNs reduces off-target activity by reducing both proteins to bind nearby sites, and delivery strategies are being optimized. Future directions in gene therapy with ZFNs. Integration with other gene editing tools, known as the CRISPR-Cas systems, personalized medicine tailoring ZFNs to individual mutations, potential clinical applications and trials, further research needed on safety and delivery, use in personalized medicine to target patient-specific mutations, combining ZFNs with other technologies like CRISPR, CRISPR for improved outcomes, expanding clinical trials to test safety and efficacy in humans. In conclusion, ZFNs offer a powerful tool for precise gene editing in therapeutic contexts. Early studies show promise, but challenges in specificity and delivery remain. Future work will focus on refining these tools and exploring clinical applications.